Yamanaka. So all you graduate students out there, you can imagine, I mean, this is an incredible accomplishment for one person. Anyway, he was working in Japan, and the approach they took, based on all this information that I've given you, had to do with the idea of there are clearly factors, proteins, genes that encode these proteins, that are controlling which genes are on and off in an embryo. And if you want to make a cell look like an embryonic stem cell, which genes are found in these embryonic stem cells? And so they studied the literature, because lots of people had been working with these stem cell lines. So I already described to you the stem cell lines that Thomson made, embryonic human stem cell lines, mouse stem cell lines. People could study these and find out what genes are present in these stem cells that are not present in other kinds of cells. And so Yamanaka came up with a list of 24 candidate genes. And the idea was, these are all genes that are known to be expressed only in stem cells. Can we put these genes back into a skin cell or a fibroblast and turn it into a stem cell? And so they actually did this with viral vectors that were able to infect cells. They did all 24 of them at once, and they showed that they could indeed change a fibroblast into something that looked like a stem cell. But that's a very crude experiment. You throw 24 genes in, and you don't really know which of those 24 are the important ones. So they started eliminating single genes. So they say instead of putting in 24, they put in 23. And they looked for the experimental case where these cells were no longer formed into embryonic-looking cells. And they found 4 genes that when they took that one, each one individually, out of the mix, it didn't work anymore. And so what